do. For example, if you deprive yourself of hunger and you develop a protein hunger or essential, and I think the difference is that proteins consist of essential amino acids. There's just this, I forget if it's 9, I think, amino acids that your body cannot synthesize. So you absolutely need them or you will die. And so, whereas sugar and fat can be interchanged with other macronutrients. So, and there's other things also that you absolutely need to ingest, like sodium chloride, right? So sodium. So there's if you deprive an animal of sodium, they'll develop this salt appetite that's incredible, basically, and that's completely innate. But that's, I think, salt appetite and protein appetite are the things that are probably the most strongly regulated at the level of the macro and micronutrients.
I'd like to take a brief break and acknowledge one of our sponsors, Element. Element is an electrolyte drink that has everything you need. That means the electrolytes, sodium, magnesium, and potassium in the correct amounts and ratios, and nothing you don't, which means no sugar. Now, I and others on this podcast have talked about the critical importance of hydration for proper brain and body functioning. Even a slight degree of dehydration can diminish cognitive and physical performance. It's also important that you get adequate electrolytes. The electrolytes, sodium, magnesium, and potassium, are critical for the functioning of all the cells in your body, especially your neurons, your nerve cells. Drinking Element dissolved in water makes it very easy to ensure that you're getting adequate hydration and adequate electrolytes. To make sure I'm getting proper amounts of hydration and electrolytes, I dissolve 1 packet of Element in about 16 to 32 ounces of water when I wake up in the morning, and I drink that basically first thing in the morning. I'll also drink Element dissolved in water during any kind of physical exercise I'm doing, especially on hot days when I'm sweating a lot, losing water and electrolytes. They have a bunch of different great tasting flavors of Element. My favorite is the watermelon, although I confess I also like the raspberry and the citrus. Basically, I like all the flavors. And Element has also just released a new line of canned sparkling Element. So these aren't the packets you dissolve in water. These are cans of Element that you crack open like any other canned drink, like a soda, but you're getting your hydration and your electrolytes with no sugar. If you'd like to try Element, you can go to drinkelement, spelled LMNT.com/Huberman to claim a free Element sample pack with the purchase of any Element drink mix. Again, that's drinkelement.com/Huberman to claim a free sample pack. If we could talk about body weight homeostasis for a moment, I think that would be useful. So let's say somebody decides they want to lose some weight, they caloric restrict slightly, either by exercising more or eating less or both. Their body weight drops by a bit. Let's say they lose 10 pounds, 8 of which are body fat. They lose a little bit of lean mass also. They're now at a new lower body weight. Are the AGRP neurons motivated to have them seek out more food? In other words, are they hungrier and more motivated to find and eat food? Or do these AGRP neurons learn, hey, body weight is lower and I don't need to push to find so much food so often?
No, I mean, the idea is that the HRP neurons are more active when you lose weight, and that chronic activation of those neurons, in part because leptin levels are lower in the blood because you've lost weight, is that drive, that counter-regulatory drive that drives you to then consume more food.
But then how do people ever keep weight off?
Well, so part of the answer is they don't. I mean, so there's
Really? Because I would argue, like, I have these friends who were very heavy. Most of the excess weight was body fat for a long time. They seem to be doing great, eating the way that I described before. And by the way, I'm not a proponent of any one particular diet. I have vegan friends, carnivore friends, et cetera. But that pattern of eating I described before has been enormously successful for them. I haven't run a randomized controlled trial. It's not my job to do that in the realm of nutrition. But they're doing great. They claim to be sated. They are so happy with the way things are going. And I don't hear that they're constantly hungry. I hear that they're constantly sated.
Well, so I would say that there have been efforts for a long time to develop diets that would help people consistently lose weight, and it has been very unsuccessful. There are some people who, for various reasons, can successfully lose weight and keep it off, and I don't know that I have a good answer for what's going on in those individual cases, how they are the exceptions to the rule. What about them is different that makes them
Some also quit drinking alcohol.
Yeah, so there's other things.
Right.
So, I think
so behavioral regulation is better when you're sober as opposed to
you can change your environment.
Yeah.
But, so what this is sort of getting at is what is the counter-regulatory response to weight loss? And so this has been studied. It was first studied in the context of energy expenditure. And because energy expenditure is actually surprisingly easier to measure in humans than food intake, because people don't tell you accurately what food they eat. If they're free-living humans, they have to fill out a questionnaire. But And the idea is that for every kilogram of weight you lose, so it's about 2.2 pounds, I think, your energy expenditure decreases by about 30 kilocalories a day. Now, so not a ton, but that is significant, right? 30 calories. And then if you lose, as you said, 10 pounds, then that's 150 calories, and that adds up over time. One interesting thing about that is that if you take people who were obese and then they've lost a ton of weight, so there's a study by Rudy Leibel about 25 years ago that did this, take people who lost like 100 pounds, and then take a control group that has the same height, weight, basically the same body composition as those people who've now lost 100 pounds. Compare their energy expenditure. The energy expenditure in the people that lost all the weight is about 25% lower than the people who never were obese. And so those people who lost the weight, we call them the reduced obese, or that's what they were called in those studies. And the idea is that there's this chronic deficit. They have to eat 25% less than someone who looks the same as them, is the same height as them, the same weight as them, in order to maintain that body weight. What's unclear is whether that's because those people simply always had a slower metabolism. They were always destined to be obese, and then you're comparing two different groups, or whether something about the process of gaining weight and being in a higher weight for a longer period of time changes the brain so that then once you lose the weight, it's irreversible. But there have been studies looking at least a year, and it doesn't seem to come back within a year, that difference in energy expenditure. Now, a question is that really the big effect? Is that why it's so hard to lose weight, energy expenditure, or is it because you're hungrier? And that's actually much harder to measure. But there was another really nice study, again by Kevin Hall, investigating this. Used a really clever approach, this drug. So basically what he wanted to do is he reasoned that you can measure people's body weight and you can measure people's energy expenditure. And because calories in, calories out, if we can measure body weight and energy expenditure accurately, we can then back calculate how much that person was actually eating. So let's see what happens when you have people lose weight. How does their food intake change? But the trick to this is you need to do it in such a way that you don't just tell them to go run on a treadmill. Because if you tell someone to go run on a treadmill and lose weight, then basically there's all these, they're thinking about the fact that they're doing this. So you need to do it in some way covertly so that you increase their energy expenditure, cause them to lose weight, but without them realizing that's what's happening. So they gave them these drugs, these SGLT2 inhibitors. And it's a pill you can take. They're used for diabetes. They block this protein SGLT2 in the kidney that is necessary for glucose to be reabsorbed into the blood. And so basically what happens is you pee out about like 90 grams of glucose a day. But you don't know that you're doing that. And that causes you to lose energy. And so these people would lose some weight. And then measure how their food intake changes. And what that showed is that for every 2 pounds or so of weight you lose, your hunger goes up by 100 calories per day. So basically you've got a 30 kilocalorie decrease in energy expenditure, 100 kilocalorie decrease in appetite for every 2 pounds you lose, on average. Some people will be exceptions, right? And they won't experience that at all for aspects of their physiology we don't understand. And so the increased hunger seems to be the main reason people find it so difficult to keep weight off.
That seems the perfect segue to talk about GLP-1, glucagon-like peptide 1, Ozempic, Mounjaro, and similar drugs. My understanding of the back history on these is that a biologist obsessed with Gila monsters, a reptile that doesn't need to eat very often, discovered a peptide within their bloodstream called extendin that allowed them to eat very seldom. It curbed appetite in the Gila monster, of all things. And it has an analog homolog, we don't know. I don't know the sequence homology exactly, but there's a similar peptide made in mice and in humans that suppresses appetite. If you would, could you tell us what is known about how GLP-1 works to suppress appetite, where in the body and or brain?
Sure.
And your sort of read of these drugs and what's happening there, good, bad, exciting, ugly.
Sure. Be happy to.
Anything else?
So the story of GLP-1, so the Gila monster is an important turn, and I'll talk about that. It actually goes back before that, quite a ways. So I should take a step back and say, these were developed as drugs for diabetes, right? And diabetes is a condition where basically you have elevated blood glucose, either because you don't produce enough insulin or because your insulin is not effective. And so back in sort of the 1920s, right around the time insulin was discovered, there was this phenomenon discovered known as the incretin effect. What it was
Incretin?
incretin,
Not the cretin effect.
not the cretin
You
effect.
can observe the cretin effect in numerous places in daily life and online. Just kidding.
So it's called the incretin effect. You can think of it as increase insulin because that's what the effect is. And the idea was that if you take glucose by mouth, if you consume glucose orally, versus if you have the same amount of glucose injected intravenously, more insulin is produced when you take the glucose orally versus if it's delivered intravenously, suggesting something about the process of ingesting the glucose causes more insulin to be released and causes you to lower your body sugar more accurately and more strongly.
Interesting.
Which is a little bit counterintuitive because in the pancreas, right? So insulin is released from the pancreas, from the beta cell. The pancreas senses the glucose concentration in the blood directly. And so it suggests that insulin is being released not just in response to changes in blood glucose, but in response to a second factor. And so they called that an incretin. And through various experiments, it was shown that this incretin effect comes from the intestine, that there's some substance being produced by the intestine that when you eat a meal, if sugar goes through your intestine, that boosts this insulin response to glucose in the blood. And people immediately realized this could potentially be very valuable. And the reason is that, you can treat diabetes with insulin injections, but insulin is dangerous, right? Because if you inject too much insulin, you can kill yourself by making yourself hypoglycemic, right? So you have to be very careful. But the thing about the incretin effect is it's not causing insulin release directly, but it's rather boosting the natural insulin release that comes when your glucose is higher in your blood. So it's sort of an amplifier on the natural insulin release. So basically, in the years that followed, whenever someone would find a new hormone, they would test it. Is it this incretin? And there's lots of failures. They weren't the incretin. But then, so there's this other hormone that comes from the pancreas called glucagon, right? And so glucagon was also discovered in the 1920s. Glucagon is kind of the anti-insulin. So when blood sugar goes low, glucagon is released in order to cause your liver to release glucose into the blood. So glucagon and insulin are these two opposing hormones. Glucagon was known for a long time, but people discovered in sort of the 1980s that the glucagon gene is expressed in other tissues other than the pancreas, and it's differentially processed. The protein is differentially processed to produce different hormones other than glucagon. And they discovered there was one in the intestine, and so they called it glucagon-like peptide because it comes from the same gene, but it's just slightly different. It's cut up slightly differently. And this hormone was an incretin. So basically, if you put it on beta cells, you get this increased response of insulin in response to glucose. And so there was the idea, okay, this could be a great diabetes drug, right? And I should say there was one other incretin that's been found. It's called GIP, G-I-P, and that will be important to talking about some of these other drugs. Also a hormone that comes from the intestine. And so the challenge with making GLP-1 into a drug is that it has an extremely short half-life. So it has a half-life of about 2 minutes in the blood. And so even if you inject people with GLP-1, it won't really be useful for anything. You don't decrease appetite, you don't affect blood sugar because it's just degraded too fast. And the reason it's degraded is because there's an enzyme, DPP4 is what it's called, that degrades GLP-1. So the first thing people tried was let's make inhibitors of that enzyme so we can boost this natural GLP-1 signal. And those are approved diabetes drugs. They're called gliptins. You've probably heard about them. Januvia is the most common one. And those boost the level of GLP-1, the natural GLP-1 in produced from the intestine by about threefold. And they're effective in treating diabetes.
People lose weight?
People do not lose weight.
Interesting.
And that's one of the key reasons that we know that the natural function of GLP-1 is not really to control body weight, because you can boost the level threefold with these DPP-4 drugs. Millions of people have taken them. They do not lose weight. That's a great question. So, but a threefold is great, but you'd like to increase it even more, right? And to do that, you can't block this enzyme. You have to actually produce a GLP-1 that is more stable in the blood. And that's where this lizard that you mentioned comes into play. It produces a stabilized form of GLP-1 in its venom. No one knows why. One hypothesis is that it's something to do with the lizard, as you said, basically having this long time period between meals and it needs to regulate its blood glucose. Who knows if that is true? But it turned out to be fortuitous because then this GLP-1 from this lizard, it has a half-life of 2 hours. And so the first GLP-1 drug that was approved was just this molecule from this lizard, basically. And it's called exenatide, and it's approved in 2005. Works well for diabetes, has a half-life of 2 hours. You inject it, and doesn't cause a ton of weight loss. But 2 hours is good, but it's not so great. So then the pharmaceutical industry said, can we, basically improve this even further? And so they start engineering this hormone, making mutations, attaching lipid tails to make it bind to proteins in the blood that would stabilize it.
Chemistry jockey stuff.
Yeah, exactly. Yeah. And I think the next big advance was this compound liraglutide. And liraglutide was approved for diabetes in 2010 and then for weight loss in 2014. And so liraglutide has a half-life of about 13 hours in the blood. So now you're getting up to something serious. We've gone from 2 minutes, 2 hours, 13 hours. And you get better effects on aspects of blood glucose and diabetes control. And they started to see that some people were losing weight. Very variable responses. Not everyone loses weight on liraglutide. And one of the things they noticed that I think is fascinating, just sort of example of how drug discovery works in the real world. A lot of these people who take liraglutide, now it has this longer half-life, they'll start to get nauseous. And that would limit how much of the liraglutide they could take. And it's a known side effect of these GLP-1 drugs. It causes nausea and sort of this gastrointestinal distress. But they noticed that over time, the nausea would just sort of go away. And so they would start dose escalating, sort of raising the dose that the person would take. So you would go, a month at this dose, and then a month at a slightly higher dose, and then a month at a slightly higher dose. And you could work your way up. And these side effects would reappear, but then they go away. And then once you got up to the highest doses, then people really started losing weight. And so there's a couple of things there. The pharmaceutical industry realized, wow, these are potentially really effective weight loss drugs. And also this nausea, which we thought was a killer, people are able to just get used to it and then it just goes away. It undergoes, the word is tachyphylaxis. So the idea is that the receptor that's affecting in the gut that's causing these effects, it undergoes some sort of downregulation with chronic exposure. So liraglutide, it's been around, it's been on the market for 14 years now. Was used, but still you're only getting sort of like 7 to 10% weight loss, which is good, but not like, amazing, impressive. But then semaglutide came along, and that was approved for diabetes in 2017. And semaglutide is Ozempic, or also marketed as Wegovy for weight loss. And semaglutide now has a half-life of 7 days. So now we've gone from 2 minutes, 2 hours, 13 hours, 7 days. And you can really jack up the concentration with a 7-day half-life. And then they saw people start really losing weight. And so in some of those trials, people lost 16% of their body weight, which previously had been unattainable. Right?
In what time frame?
Typically takes about a year.
Okay. So, and most of the loss in body weight is from body fat or from other compartments?
The typical number is that if you lose weight either through dieting or through taking one of these drugs, and you don't do anything like eat a high protein diet or do resistance training, somewhere between 25 and 33% of what you lose is going to be muscle. The rest is going to be fat.
But as you said, some of that could be offset by resistance training and or consuming a higher protein diet.
Yeah, you can almost completely eliminate that if you eat enough protein and do serious weightlifting. Obviously, not the whole population is interested in doing that. And there's been a lot of discussion of how serious a side effect this is. Among elderly people, you don't want to be losing muscle mass because you're already losing so much muscle mass. On the other hand, the counter argument that has been made, which I think is also kind of convincing, is that true, you're losing some muscle, but you're also losing all this fat, and you no longer need as much muscle when you're not carrying around as much body fat. So people who are heavier naturally have more muscle because they need to move their body, right? And so
the calves on very obese people are often enormous.
Exactly.
And then they lose weight and
exactly.
And I mentioned the calves in particular because they're carrying a lot of the body load.
Exactly. So it's still an open question as to how serious a problem this lean muscle mass loss is, although the pharmaceutical industry is all in now on making drugs that basically are going to prevent that. So that's something that will be happening probably in the future.
Is it sorry to interrupt, but is the weight loss on these drugs the consequence of reduced appetite or some other aspect of metabolism? And if it's the consequence of reduced appetite, is that occurring at the level of the brain and gut? Or a combination?
So it's almost entirely reduced appetite, and it's almost entirely occurring at the level of the brain.
Which neurons?
It's thought that the key targets of these drugs are neurons in these two regions. One's called the nucleus of the solitary tract, and the other one's called the area postrema.
So we're back in the brainstem.
Back in the brainstem. So these are actually the neurons in that decerebrate rat story I was telling earlier. These are the brain regions that are preserved in the decerebrate rat. The decerebrate rat still has these very caudal brainstem structures. They're two very special brain regions because they get direct input from the vagus nerve. So the vagus nerve is the nerve that innervates your stomach and intestines and heart and lungs. And it's sort of the major pathway from gut to brain. It provides most of the the neural input from gut to brain, telling you about things like this, your stomach distension, how many nutrients are in your intestine, breathing, all that stuff. And almost all of those vagal nerves terminate on these two structures in the brainstem.
When I hear postrema, I think about nausea because I was taught that postrema contains neurons that can stimulate vomiting. And this seems to link up well, at least in the logical sense, with the idea that stimulating, activating receptors in these neurons within postrema might explain part of the transient nausea side effect of Ozempic and related drugs.
Yeah, so the current thought is that a lot of the nausea is coming from activating the neurons in the area postrema, and that a lot of the sort of physiologic satiety is coming from activating the neurons in the nucleus of the solitary tract. Now, the whole brain is connected to each other, and so if you really turn on these neurons in the NTS and the AP, they're going to talk to the hypothalamus and all these other brain regions. It's going to change the whole brain. So it's not just those regions, but these drugs don't have great access to the brain. They can penetrate a little bit into the brain, but they don't penetrate into the whole brain. And it's thought that if you take fluorescently labeled versions of these drugs and see so you can visualize where do they actually go, they're enriched in these structures in the brainstem. So that's why people think that this is probably where they're acting.
And is that because there's an abundance of the receptors for these compounds in postrema and NTS, or is it because the blood-brain barrier is somehow weaker at that location?
It's because the blood-brain barrier is weaker. So basically, it's a region, what's known as a circumventricular organ, meaning it's one of these rare places in the brain where the blood-brain barrier is weakened, and so substances can come from the outside into the brain. And that's important for these big peptides because these are not small molecules. These are big peptides with lipid chains on them and other things. And so they can really only get into areas of the brain where the blood-brain barrier is weakened.
I really appreciate that you mentioned the half-life issue with GLP-1 and the fact that these DPP-4 antagonists did not lead to weight loss despite increasing circulating GLP by threefold. This is relevant to a number of different claims that people make that a given food or a given drink increases GLP-1. Yeah. I've actually said before, I'm a big consumer of yerba mate. My father's side is Argentine, and it's a known appetite suppressant, but it contains caffeine and other stimulants that might explain some of that. And it's not a robust appetite suppressant to the point where most people would, rely on it as a weight loss compound. But anyway, it's my preferred source of caffeine, but I've said before, there's some evidence that it can increase GLP-1, but based on what you've said, the increases in GLP-1 that it creates are very unlikely to produce the kind of appetite suppressive effect that would lead to any significant weight loss in somebody that's obese, presumably that are separate from any caffeine stimulatory effects, right? Yeah. So you can't separate because it's a complex compound, this yerba mate thing. It's got lots of things in it. But also, I've observed you being vocal on social media when people have said, hey, this thing increases GLP-1. You quite appropriately, I think, said, wait, Ozempic and drugs like that increase GLP-1 1,000-fold. When you talk about a food or drink, or maybe a supplement increasing GLP-1, it's very unlikely to increases GLP-1 to that level, meaning unless you're getting into the 100-fold or 1,000-fold increases, probably not right to talk about GLP-1 being the source of any appetite suppressive effect.
Yeah, that's all correct. So, I think it's important sometimes to distinguish between pharmacologic and physiologic effects. So, physiologic is what the hormone naturally does in your body and what can be modulated by natural things like eating a different food. And you might get a twofold change in your GLP-1 by eating a different food, one food versus the other. But as we know from those DPP-4 inhibitors, it's not going to really change your appetite because the drugs increase it threefold. These GLP-1 agonists are really a pharmacologic effect that only happens with drugs. So you get 1,000 to 10,000-fold higher concentrations of these drugs in your blood than the natural hormone. And so it's just, there's no diet that's ever going to give you that.
And there's no precedent for it either. So should we be at all concerned about that? They run clinical trials and address safety, but when you're talking about 1,000-fold increase in essentially a peptide hormone, if we were talking about a different peptide hormone, pick one, oxytocin or estrogen, testosterone, they're not really broadly speaking, most people would be concerned about 1,000-fold dosing of something like that. And obviously there are clinical indications where that's important. However, my observation of the ever-expanding literature on GLP-1 agonists is that there seems to be improvements in like reduction in alcohol consumption. And by the way, why would increasing GLP-1 reduce craving for alcohol? That it seems like there's an ever-expanding list of things that GLP-1 agonism is good for.
Yeah.
But we are talking about, I would say, supraphysiological levels
Absolutely.
when one takes it. And again, I'm not against it nor for it. I'm just paying attention to the literature.
So I would say that that's absolutely right. When you're increasing the level of a hormone 1,000-fold, you need to be careful, see what's happening. But at the end, it's an empirical question. What does it actually do to a person? And it can only be answered through experiments. And I think the nice thing about these GLP-1 drugs that a lot of people don't realize is they've been around, approved since 2005, the earliest ones. And even something like Ozempic, which maybe only entered the public consciousness in the last year or two, right? It's been around for 7-ish years, I think. So, and big clinical trials with these drugs. And so, and the evidence so far is that they seem to be incredibly safe. And as you said, not just incredibly safe, but they seem to have all these unexpected health benefits that seem to be in some cases even unrelated to weight loss. And so because of the reasons you mentioned, one of the things the FDA requires from these pharmaceutical companies for diabetes drugs is these large cardiac outcome trials. So basically where you measure stroke and where you measure heart attacks and death from any cardiac cause. Big trials, like 20,000 people, 4 years, cost like $1 billion to run. And the data from the semaglutide, the Ozempic trial, came out last year. And as expected, reduced the rate of heart attacks, strokes, all-cause mortality according to cardiac for cardiac reasons. But what's really surprising was a lot of that seemed to happen before the people even lost weight. So there was already a difference between the placebo group and the semaglutide group before the people on the drug had lost a significant amount of weight. And there was no correlation between the amount of weight they lost and how well they were protected from heart disease. And that's led many people to think that some of these effects actually could be due to other things the GLP-1s are doing that we didn't expect. And so one thing is there's an idea emerging that they are anti-inflammatory. So these brain regions, the area postrema and the NTS, are also really important for this reflex known as the inflammatory reflex that basically acts, it starts with the vagus nerve, goes to these brain regions in the brainstem, and then goes back down to the body to basically to prevent out-of-control inflammation. And so it's thought that these drugs perhaps have an anti-inflammatory effect that explains some of that.
Sounds like the patent on these drugs just got extended by another 100 years. That's a bio pharma joke. I mean, just to put context on it, drugs can be patented and sold as a commercial version and not as generic versions until the patent runs out, unless companies are able to find another approved clinical use, in which case it can be remarketed only as a brand name, not generic version. So a lot of companies, once they do the safety testing and all, given everything they put into the R&D, into the research and development, there's a very big incentive to not necessarily finding new drugs, but finding new uses for the same drugs. Absolutely. And not allowing generic versions into the picture. Yeah, and that's why it's likely to be, based on these what sounds like additional uses of Ozempic-related compounds, a long time before there's generic Ozempic available.
I think it will be a while. I don't know the exact status of the patents, but I'm guessing it's going to be a while before there are generic versions, but there's a lot of competition coming. So every major pharmaceutical company, or almost every major pharmaceutical company, now has a GLP-1 program. Really? And some of them are really exciting, actually. So I mean, the general trend in this area is what people call GLP-1 plus, which means you take the GLP-1 agonist, which is already giving you 15% weight loss or so, and then you add additional things to that to give it additional properties. So one compound is from Eli Lilly, which makes so there's this other drug on the market that we haven't talked about, but tirzepatide, which is known as Mounjaro for diabetes, and Zepbound for obesity, which is even better, really in almost every respect, a better drug than Ozempic. So people lose more weight. So it's about 21% weight loss at a year. Fewer side effects, at least at comparable doses. that seems to be because this other drug, tirzepatide, it has two targets, not one. So whereas Ozempic is just GLP-1 receptor agonist, tirzepatide is a dual agonist of GLP-1 and this other incretin that we talked about, GIP. And it seems like having that GIP agonism actually acts as an anti-nausea effect that sort of counteracts some of the nausea caused by the GLP-1. And in the area postrema, there are GIP receptor neurons in the area postrema, this nausea center. So it allows you to crank up the dose of the GLP-1 agonism even further while you're suppressing the nausea and just get even more weight loss. So now, talking about the future, things that aren't available yet but will be in the next couple of years. So Eli Lilly, the company that makes this drug, tirzepatide / Mounjaro, they have a triple agonist that's in phase 3 clinical trials now. So this is now 3 hormones in one. It's the GLP-1, which all these drugs have, the GIP, which is the anti-nausea component, and then glucagon itself. And so these 3 hormones all combined in one pill. And what the glucagon does is it increases energy expenditure. And this is a well-known effect of glucagon. And so you're basically eating less, your nausea isn't as bad, and now you're just burning more calories at baseline. And the results from this drug are incredible. So basically, there's been one phase 2 trial published, and people lost 25% of their body weight at the end of the I think it was 48-week period, and they were still losing weight. So we don't know where the end point. We don't know what the maximum is. So there are bigger, longer trials going on now to figure that out. But at that point, when you get beyond 25% body weight, you're talking about basically bariatric surgery, right? Which is currently the best thing we have. Like these surgeries people do to
quote unquote stomach staple, removing a portion of the stomach,
removing a portion of the gut. So really, it's a pharmacologic version of bariatric surgery. The other one that I think is really exciting, there's this compound from Amgen. It's called, just right now it's just a code. It's like AMG 133. But it's like tirzepatide in the sense that it targets both GLP-1 and GIP. So it's a dual targeted. But unlike tirzepatide, which activates the GIP receptor, this Amgen compound inhibits it. And for reasons that people don't understand, either activating or inhibiting this receptor causes you to lose weight. So it's still a mystery, but a lot of debate about what's going on there. But the way this Amgen compound activates the GIP receptor, or inhibits the GIP receptor rather, is that it's an antibody. So all these other things were peptides, but this is a much bigger, it's actual protein. This is an antibody. And because it's an antibody, it has a much longer lifetime, even than something like semaglutide, which is 7 days. So it lasts like a month in the blood or something. And so you give people monthly injections of this, and they lose dramatic amounts of weight. And then at least in this initial trial, at the end of this, they stopped, and people maintained the weight loss for 6 months.
That's impressive.
Potentially because of the long-lasting effects of this antibody, or potentially because of other things that we don't understand. So, and those are just 2. There's all sorts of other crazy things happening. So really, I think it's just created this explosion of interest in pharma. Basically, it's one of these things, it's once you see that something can be done, all of a sudden that changes everyone's perspective. And so now obesity drug discovery has gone from something that 10 years ago everyone wanted to stay away from because there were so many nightmare stories about drugs that turned out to be not safe, till now everybody's sort of all in on this.
Yeah, I remember in college the fen-phen debacle where a diet drug was released and people had cardiac issues, started dying, so it was pulled from market, and then it was essentially a quiet field for a long time. In part to bring us back into the brain, and in part because it's directly relevant to what we've been discussing about Ozempic and GLP-1. There are other neurons in the brain that regulate feeding, and there are other peptides involved in appetite control, for which I would say niche communities have started to indulge in. And by the way, people were taking GLP-1 analogs long before they were FDA approved in kind of niche communities. These aren't communities I'm a part of, but every once in a while I'll stick an ear into one of these communities and hear what people are taking. And a big thing right now in these communities is the use of other peptides that are in the melanocyte stimulating hormone pathway. And you mentioned melanocortin receptor containing neurons. Could you tell us a little bit about what these neurons do in the absence of any pharmacologic stimulation, and then why it would be that people would perhaps stimulate these pathways with these drugs? Not that we're recommending that,
Yes.
but I do think that given that some of these neurons are also involved in sexual behavior and FDA approved for the treatment of hyposexual function in women, things like that. There is FDA approval for some of these compounds that they're interesting hypothalamic neurons that are starting to gain more attention, and that I predict, based on their potential involvement in feeding appetite and weight control, are likely to enter the picture with more prominence in the not too distant future.
So alpha MSH, as scientists call it, the hormone you were just referring to, is a product of the POMC gene. So in the same way that we just talked about how glucagon can be processed into different things, in some cells it's made into the glucagon hormone, in other cells it's made into GLP-1. POMC, that gene can be processed to produce different hormones, and one is alpha MSH, which is very important for feeding control. And so these POMC neurons, they're in the arcuate nucleus of the hypothalamus, the same region where these AGRP neurons I talked about earlier are located. And there's sort of these two sets of neurons have opposing effects on body weight regulation. And so alpha MSH inhibits food intake, and AGRP neurons promote food intake. And where they converge is at this receptor, the melanocortin-4 receptor, which is important for body weight regulation. And so alpha MSH is an agonist; it turns on that receptor, and the AGRP peptide is an antagonist; it turns it off. And so, there's a lot of human genetics, as I mentioned earlier, implicating this pathway in body weight regulation. There have been a lot of efforts over many years to turn alpha MSH into a drug, and it's been very difficult. There is one drug that's now approved. It's called, I think I'm going to get the name wrong, it's like setmelanotide or something like this. It's an MC4 receptor agonist. It's mainly used in relatively small populations of people that, for example, have mutations in this pathway. It's not used as a widespread as a drug. And the challenge has been really side effects. So there's an increase in blood pressure that happens sometimes with these medicines, partly because this pathway controls not only appetite but also autonomic tone and sympathetic nervous system activation. So it's just taking a step back from everything we've talked about today. I talked about how there's this short-term system and the long-term system that controls energy balance and body weight. The short-term system in the brainstem, the long-term system in the hypothalamus, the long-term system being leptin and alpha MSH and AGRP. When I was coming up, learning about this stuff 15 years ago or 20 years ago, it was the dogma was you could only affect body weight through the long-term system, by manipulating the long-term system, because any manipulation you did of the short-term system in the brainstem, the animal would just compensate. And there were these famous experiments where they would take CCK, which is a hormone just like GLP-1, inject it into rats, inject it several times a day. And CCK is known to decrease the size of meals, and it would decrease the size of meals, but the rats would never lose any weight because they would just eat more meals to compensate, and they would just perfectly compensate by eating more meals. And so the lore was, it's just impossible. The animal will always compensate unless you hit this body weight set point regulating area, which is the hypothalamus, the long-term system. But then what the pharmaceutical industry discovered, which I guess maybe shouldn't be so surprising, but I guess it was to some people, is that if you just hit that receptor, that short-term system, 24 hours a day, 7 days a week, and never let it stop, then you will lose weight, right? And so the short-term system alone is enough to cause body weight regulation. On the other hand, the long-term system with alpha MSH and AGRP neurons and POMC and all this stuff has been a challenge to pharmaceutically target. As you know, leptin, we discussed, didn't really work. And so I think there's going to be, as you mentioned, a reemergence of interest in considering this other pathway now that we've seen the success of the GLP-1s. And I think one area where it may emerge is in considering their combination, perhaps at different stages of weight loss. So perhaps, what would make a lot of sense scientifically, I don't know if it'll work in practice, is that you would take a GLP-1 drug to lose the weight. And then at some point you might stop that drug and switch to a more hypothalamus-centered leptin-based drug to keep the weight off. So basically, use the GLP-1 drug to force yourself to lose the weight, and then use the leptin hypothalamus-based drug to sort of say, okay, this is our new body weight set point. Let's not resist this weight loss that's happened. Whether that will actually make sense practically is hard to say because, the GLP-1 drugs have just a lot of benefits, even beyond weight loss, so people might not want to stop taking them. But that's one idea.
Very interesting. I'd love to talk about dopamine.
Sure.
We hear so much about dopamine being involved in pleasure. I like to think I've had at least a small level of impact in convincing people that it's also involved in, perhaps mostly involved in, things like motivation, different forms of learning, and lots of other things too, folks. Dopamine does lots of things. It's even expressed in the eye and controls adaptation to light. So it does lots of things, but it certainly is believed that dopamine is involved in our either craving for food or pleasure from food. What's the real story on dopamine as it relates to food and eating behavior? You had a beautiful paper published in Nature entitled, and we'll put a link to this in the show note captions, Dopamine Subsystems That Track Internal States. And I love this paper for a variety of reasons. But if you could give us the high points of your discoveries on dopamine as it relates to feeding, I think I know, in fact, that people would find it very illuminating.
Sure, fantastic. The question of what dopamine does with respect to feeding is a great question and a difficult question, I think, to answer. There's a lot of misconceptions. I think the evidence is dopamine probably isn't so much involved in the pleasure of food, that taste, the hedonic experience. One reason we think this is because you can make mice, Richard Palmer did this decades ago, that don't have any dopamine, and they still show the same sort of effective responses to food. So you put something sweet in their mouth, they like it, right? What dopamine seems to be important for with respect to food is two things. One is the motivation to engage in work to get food, particularly when it's high levels of effort. So if you ask a mouse to press a lever to get a pellet of food, if it doesn't have any dopamine, it won't do it. And if it has low levels of dopamine, it'll just work a little bit. So dopamine is important for sort of energizing action and motivating you to engage in hard tasks. The other thing that dopamine is really important for is learning. And it's important for learning about which cues predict something useful for the body. And feeding is a central example of that. And what that paper of ours is about is the idea that this learning actually happens on two different time scales for two different kinds of cues. So what we almost always talk about with dopamine and learning, which is important, is learning about how external cues in the environment predict something like food availability, right? So you see a McDonald's sign and you know that that means there's some tasty food in there. And so dopamine is involved in that process of sort of learning what that external cue means. And that's a very fast timescale process. So in the laboratory, for example, we will play a tone and then give an animal a sip of a solution that has calories in it, for example. And it can learn the association between that tone and that the food is going to be available if they're separated by a few seconds, but that's all. And that's a dopamine-dependent process. But there's a second sort of much slower timescale learning about food, which isn't about where I go to get a hamburger, but rather about what the experience of eating the food, the orosensory experience, its taste, its flavor, its texture, how that relates to the post-ingestive effects.
And I should say that this seems extremely relevant to the McDonald's example because in your experimental situation, the tone is analogous to the golden arches of the McDonald's sign.
Exactly.
But in my experience, and forgive me, but most of the food that I've consumed from McDonald's does not taste good relative to other like really delicious hamburgers or French fries or something like that. I mean, it's so you're saying dopamine is required to link the signal, the golden arches or the tone, to the presence of food at a particular location.
Exactly.
But not to the experience of pleasure from that food.
Exactly.
Which squares very well with my experience of McDonald's. And I probably haven't had a bite of McDonald's in 20 plus years.
Yes.
I would have to be pretty hungry.
I haven't either. And it's funny, the golden arches thing is just something that people in neuroscience talks about dopamine use. And so now I've started subconsciously just talking about golden arches, even though I also haven't eaten at McDonald's in decades.
Yeah. In-N-Out burger, better tasting, from what I understand, probably better sourcing. We're not going to get into all of this in detail, but everyone has their preferences. But I do think it's interesting because what we're talking about here is related, I think, to this notion of highly processed food, packaging, the commoditization of food, just the idea that we are drawn to food for things other than the taste that we expect for it. There's all this context.
That's right. So I think an important distinction that people make is the distinction between wanting and liking. I don't know if you've talked about this previously on the podcast.
Anna Lemke, my colleague at Stanford, came on the podcast, talked about dopamine is about wanting as opposed to enjoying.
Exactly. So
in most cases.
Yeah. So liking is the subjective hedonic pleasure in the moment of eating it, but wanting is just what you want. And these can be uncoupled all the time. You can want things that at the end of the day, you don't actually enjoy it when you get it. I feel like a lot of life is like that.
Indeed.
And so dopamine is very powerful at making you want something, but not necessarily like it. So that's one element. But then there's this other element that is important, but very much less studied, but I find much more interesting, which is how you connect these sensory cues associated with food, its taste, its flavor, its smell, with the consequences for the body. And this is so important because so much of whether we like or dislike a particular food or drink is related to its post-ingestive effects. You come to like things, for example, that have calories. So this is one of the reasons that adults will eat vegetables and other savory foods that children find disgusting. Even though they're a little bit bitter, you learn through experience, this makes me feel good to eat this. And even maybe at a completely subconscious level, there's also a level of learning that occurs. And this, of course, happens with other things like coffee and beer and other things like that. And so there's been an idea that this other much slower learning occurs. And the reason I say it's slower is because the time between when you taste the food and when it actually gets into your intestine and releases the hormones that might drive this is quite slow, separated by tens of minutes. But how that works hasn't been clear. There's been an idea that dopamine might be involved, but it hadn't really received a lot of attention. And so we set out to investigate what is the role of dopamine in these post-ingestive responses and sort of map out for the dopamine system, how does the dopamine system respond, not when you see the golden arches, which is usually the kinds of experiments that have been performed, but rather when you deliver nutrients directly to your stomach, or when you deliver water directly to your stomach if you're thirsty, and so on. And what we saw was that there are these different populations of dopamine neurons that are tuned to respond to signals from inside the body. And so there are some that respond when nutrients are in the stomach and intestine. There are others that respond when, in a thirsty mouse, when the blood is rehydrated, when you basically satiate your thirst. And we showed that the purpose, or at least a purpose of that activation, is to cause you to learn about the effects of what you just ate, basically to create this connection between the flavor of something and its post-ingestive effects. So that's sort of that delayed dopamine signal after ingested food and fluids is sort of reinforcing this connection between the flavor of what I just ate and that it was something good for me. One of the sort of interesting things about that paper that was not the direction we initially expected to go in is that for food, I think it's kind of intuitive. There are lots of flavors to food. You have to learn, what all these different flavors mean. For thirst, people find it a little less obvious because thirst is just water. Aren't you just born knowing what water is? Like, how do you have to learn anything to do with drinking a glass of water? But it actually is a learning question in part because for many animals, probably most animals, thirst is something that's associated with eating, not drinking. There's this study I love of rabbits in New Zealand. So there's not a lot of people studying how they get their fluids in the wild, because who cares? But it's kind of interesting. And so in New Zealand, there's this huge rabbit problem because they're an invasive pest species that was introduced in the 1800s, and they're just eating all the land. And so there's lots of money to study rabbits, to understand their ecology. And so a group of researchers did this experiment where they made this big pen outside where they put a bunch of rabbits in them. So the rabbits couldn't escape, but they had all their natural food. It was like an outdoor area. And they also put a trough of water. So the rabbits always had access to water, which is like clean water, and they could measure how much water the rabbits drank. And what they basically found is that 9 months out of a year, rabbits drink zero water. They drink absolutely zero because they get all of their water from food. The only time they drink is during the winter when all of the greenery has sort of become shriveled, and then they can't get water from that anymore. And so it's just kind of an interesting aspect of how many animals are very different from the way we think about ingestive behavior. But that fact that animals have to get water from food raises this question: how do they know which foods are rehydrating? Presumably they have to learn that, because you can't just look at a food and say, if you've never had any experience, oh yeah, this is something that's very water rich and this will rehydrate me when I'm thirsty, and this one is not. Mm-hmm. And so James, the graduate student who led this project, basically investigated this by giving mice different fluids and then measuring how the dopamine response. And he showed there was this delayed dopamine response after the mice had drank the fluids that correlated with rehydration of the blood. So a whole bunch of dopamine neurons get strongly activated when the blood is rehydrated. And he hypothesized this might be a signal, this delayed activation of dopamine neurons, that allows animals in the wild to learn that food I just ate is rehydrating. And so he did an experiment where he basically gave them two different flavors, mimicking sort of the flavors of two different foods, one of which was hydrating and one of which was not. And the animals couldn't tell because he infused the water directly into their stomach. And he showed that basically these dopamine neurons are critical for them learning that association. So that's the story of that.
I love it. And I'll tell you why. When I was in college, for reasons that I don't recall, I decided to run an experiment on myself. where I would eat one meal that was fairly low water content, like a piece of meat or something with some cheese, what some people call a keto meal, but I wasn't ketogenic. I don't even think I knew what a ketogenic diet was at that point. And then the next meal I would have like a salad and some fruits. And then I would switch back and forth. And I generally would only eat 2 or 3 times a day. Anyway, there's only so many hours in the day. And I found it to be incredibly satiating. And I found that I felt great. And I can imagine any number of different reasons for that. And there are these theories that you probably recall that the diet that was being promoted in the 90s where people would either eat carbohydrates or protein separately. Like there was some wackiness out there. Yes. And as I say that, I'm sure I'll get assaulted in the comments. It's probably not wacky. I'm sure there's some enzymatic basis for why that would be useful. If you enjoy it, go for it. I don't have a feeling about it one way or the other. But one thing I noticed was that low water content containing meals, either by virtue of the foods that they include or by virtue of the fact that they're not diluted, so to speak, it's a different taste experience to eat those foods than it is to eat like a big salad or something of that sort. Right. In any event, I don't do that any longer. I just sort of stopped, but it was a fun experiment. And I think it was efficient because at the time I had very low money as a student. So, generally fruits and vegetables were less costly than meats and things of that sort. But in all seriousness, to what extent do you think humans overeat or undereat depending on the water content of the food?
It's an interesting question. So, there is this advice that you should, if you're hungry, first drink some water and see if you're still hungry. And the idea is that perhaps humans can't always, our interoceptive sense, our ability to sense what our body needs is not perfect. And sometimes we could be confused and we could really be thirsty when we're hungry and hungry when we're thirsty. And there's some evidence that could help. I would say it's probably not a huge effect in most of modern day life, but it's an interesting idea. Mm-hmm. Yeah.
This brings us to the topic of thirst, something that your laboratory has worked on extensively, and the topic of osmolarity, of salt consumption and things of that sort. In broad terms, how do these things link up? Meaning, are there instances in which what we really need is salt and we end up eating a bunch of Parmesan cheese? I got teased yesterday by my team because occasionally when I'm on the road, I don't like most of the foods available in most airports and stuff, so I'll bring a chunk of really nice Parmesan cheese. I'll just break off a piece and eat it. I'll have half a cucumber, and I'll have a can of, not a can of tuna, but there are these wonderful jarred fillet of tunas that are available that are in olive oil. They taste really good. This is not canned tuna. It's really good. And I'd rather eat that in most cases until I can get to a decent meal than like what's put in front of me on an airplane most of the time. So I get teased about this, but I notice that, for instance, sometimes I'll eat the cheese and I think, oh, actually what I really just want is the salt.
Yeah.
I really want the salt. I've been drinking a lot of coffee today. I've had a couple extra glasses of water. Maybe I'm just craving salt and I'm confused and I'm over consuming this cheese.
Yes.
When in fact, what I'm going for is the salt. As you point out, our understanding of exactly what we need is fairly crude, and oftentimes we overshoot the margin, especially when foods are in combination. Yeah. So salt, water, and let's just say calories. How do we accurately or inaccurately pursue those at the level of biology?
Okay. So I'm
throwing tough questions at
I know,
you,
this feels,
but you're
feels like my qualifying exam. Well, there are separate systems. There's thought to be separate systems that control salt appetite, thirst for water, and hunger for calories. And so they involve different brain regions for the most part, different neurons, different signals from the body. In general, hunger and thirst are pretty separable. I would say the instance where they interact is in phenomena such as dehydration, anorexia. This is the idea that if I give you some dry food, but I don't give you any water, you're going to eat less food because basically you're going to get dehydrated and you're going to decide I need to preserve my fluid balance even if I eat less calories.
So we prioritize hydration.
Yes, you will at some
the
point.
Level of survival.
At some point you will prioritize hydration. That's related also to the concept of prandial drinking. So many animals, including humans, drink most of their water during meals because you basically want to counteract the osmolytes that are in your food. Salt balance, though, and thirst, so the thirst for water and the desire for salt are much more tightly linked because the purpose of both systems is to maintain the composition of the blood at its right concentration. So you want to have the right osmolality of the blood, which you can just think of in simple terms as sort of the total concentration of all the salts. It's a little more complicated than that, but it doesn't really matter. And you also specifically need to maintain the sodium concentration at the right level. And so, and there are really powerful innate mechanisms that drive both. I think thirst is very intuitive to people. You get dehydrated, you lose water, you become thirsty. And we know now that there are a very small set of neurons in a few brain regions that control that. And the way they're thought to work is they contain osmosensors. So they contain basically these neurons are sensors for the osmolality of the blood, and they're activated when the blood osmolality gets too high. And it's an incredibly sensitive system. So you can perceive an increase in your blood osmolality of 1% as the sensation of thirst. So remarkable.
Wow.
Yeah.
That's how critical it is to maintain salt balance.
Exactly. And so you get to 10% increase in blood osmolality and you're in extreme discomfort, and 20% you're like in the hospital.
So if I took, let's just say a half an ounce sip of seawater inadvertently.
Yes.
It's extremely aversive.
It is.
It's like, blech. Like you want to drink some non-salty water, some nice clean water.
Yes, exactly.
Immediately.
Yeah. So I should emphasize that there's two components to the fluid homeostasis system, to the water homeostasis system. One is this desire to drink, but the other is, of course, the kidney. And so the reason that drinking the salt water won't put you in a really bad situation is your kidney would then filter out a lot of that salt and cause you just to pee it out, and then you'd be fine. And so those two work in balance. The kidney's controlling how much of the salt gets reabsorbed into the blood, and then this desire for thirst, this desire to drink, allowing you to replenish the blood with water at various intervals. And so, the experiments led to the discovery of this thirst circuitry are amazing. It was this guy, Bengt Anderson, working in the 1950s. And he just had this hypothesis that there was an osmosensor in the brain, right? Which is I think, there was some evidence to suggest it, but it was not really strongly supported at the time by the data. And so he took these goats and he just started infusing small amounts of salt into various places in their brain, reasoning that if there was an osmosensor...
Sorry to chuckle. I was wild. I mean, I wasn't chuckling in jest I feel for the goats. I feel for everyone involved in that experiment. But what a wild experiment, just to put salt directly into the brain.
A concentrated saline solution, yeah.
My goodness.
And he found this tiny region in and around the hypothalamus that if you infuse salt in this region, the goats they'll drink like 8 liters of water in 5 minutes. Just crazy, right? And so he reasoned, okay, this must be the osmosensor. And then he went back and stimulated those neurons. It's just the same thing. The goat just drinks like crazy. And so now we know there's this couple small regions around the hypothalamus. One's called the subfornical organ. Another one's called, it doesn't really matter what they're called, but basically that have these osmosensors. One of the interesting things about the regulation of fluid balance is you face some of the same challenges.
We were young, wild and free, chasing dreams by the sea, echoes of a distant melody, in the lights just you and me. Photographs and shades of gold, stories that we've never told, memories that never grow old. In this heart they're always bold. Time moves on but we stay here, in the moments crystal clear, echoes of the yesteryear, in our hearts we hold them. Memories in the light, lost in whispers of the And I say, in the dawn we'll never stray. Fading memories in the light, lost in whispers of the night. In the silence we take flight, holding on to what feels right. As the days turn into years, through the laughter, through the tears, in the echoes on our steers, in these dreams we face our fears. As the days turn into years, through the laughter, through the tears, in the echoes on our steers, in these dreams we face our fears.
We just talked about with the regulation of food consumption, which is that you have this behavior, this ingestive behavior that leads to replenishment of the body, but there are these delays, right? So if you're thirsty and you drink a glass of water, it can take on the order of sort of 20 to 30 minutes for the water to be absorbed into your blood, for the blood to be rehydrated, and then for these osmosensors that Bengt Anderson discovered in your brain to be sort of sense that and return to normal activity. But of course, if you have the experience of drinking a glass of water, you know that you can quench your thirst within minutes, right? And so how does that work? Within seconds even. So one of sort of experiments we did early in my lab was to ask that question by basically recording for the first time the activity of these neurons that Bengt had discovered by putting the salt into the go. So we went back into them now in mice. Mice have the same neurons. You have the same neurons. And recording their activity when a thirsty mice drinks, it asks what happens. And what we saw was that the neurons don't wait until the blood is rehydrated. They also don't do what the AGRP neurons do, is meaning they don't look at the water and predict how much water they're going to drink. But instead, they get a signal from the mouth, which every time the mouse takes a lick of water, their activity goes down a little bit. And basically, they track in that way the volume of water that's passed through the mouth. They also get the signal from the blood, really relaying the osmolarity of the blood, and they compare these two. And basically, when the mouse has drank enough, in order for the animal to predict that the blood osmolality is going to return to normal, then the animal stops drinking.
Beautiful.
Yeah, so
It's just beautiful, right?
It's incredible.
I was like, the brain is essentially predicting with, it sounds like a high degree of accuracy, how much water one needs to drink, linking it to the pleasure and of ingesting good, clean water under conditions where we're thirsty, in anticipation of adjusting blood osmolarity in 20 minutes.
Exactly.
I mean, it's this is the kind of thing that just it delights me, because it just means that the brain as a predictive organ is so accurate.
It also explains some sort of funny aspects of thirst that you may have noticed from everyday experience. So one idea is that just cooling your mouth can sort of quench your thirst, right? So if you're in the hospital and you're not allowed to drink any fluids, they'll give you ice chips to suck on to sort of quench your thirst. So why is that? And so one idea is that perhaps because water is usually cooler than your body, that sensation of water passing, it always cools your mouth. And so you learn, or maybe it's innate, that just cooling of my mouth means that basically I'm going to be rehydrated. So this was an experiment done by a graduate student, Chris Zimmerman. Chris did the same thing where he was recording these thirst neurons, but just put a cold piece of metal on the mouse's tongue. And you can see when you do that, these thirst neurons go down in activity, and then you remove the cold piece of metal and they go back up.
Amazing.
So a lot of these sort of oddities of everyday experience have to do with how the system has evolved to make the prediction about what's going to happen to the body.
I mean, few things are as rewarding as the sensation of drinking really nice, clean, cold water when one is very thirsty. When my lab was in San Diego, I used to take my dog hiking Palomar Mountain, and one day, I really screwed up. He was a Bulldog Mastiff. They overheat easily, and it was a lot warmer than we thought. We ran out of water. It was actually dangerous situation for him. We got down to the bottom of the hill, thankfully, with him still alive, and there's this pump that pumps what is, I was told, was spring water, and it came out really cold, and you could just see him fill back up with life.
Yes.
I filled back up with life knowing he was filling back up with life. And it was unlike the kind of reward that one experiences with food when you're hungry.
Absolutely.
It's like that basic critical need for water.
Absolutely.
Under conditions where you're clearly dehydrated is like nothing else. It's delicious in a way that no food is delicious.
I would like to actually say something about this. So that distinction you made is really interesting between hunger and thirst. So when you stimulate these neurons that make an animal thirsty, the mice hate it. They will do anything to avoid something that artificially makes them thirsty. So we can artificially stimulate these thirst neurons, create a state of virtual thirst. They'll lever press hundreds of times to make it stop. The same neurons, the neurons that I talked about that control hunger, the AGRP neurons, they actually don't care so much. They won't really do much of anything to shut them off. That raises the question, well, why do the animals eat then when you stimulate the hunger neurons? And we think the primary thing that the hunger neuron stimulation does is it makes food itself more attractive. It makes the food more delicious, more of an attractive motivational magnet. It makes the experience of eating more pleasurable. But it is not itself the most unpleasant state. At least the mice aren't willing to do that much. Whereas for thirst, I think, dehydration and thirst is really just unpleasant, and animals just want to avoid that. And so I think the distinction is very real. I think there are two different motivational mechanisms for hunger and thirst. Hunger is mostly about the reward of food. Thirst is mostly about this is just really unpleasant.
And removing that unpleasant signal.
Exactly.
And you had a paper, which I was going to ask you about, so I will, entitled "The Forebrain Thirst Circuit Drives Drinking Through Negative Reinforcement."
Yes.
And I'm guessing that paper illustrates exactly the point you just made. So it's a forebrain circuit.
Yes.
Does that mean that there's some elements of learning and cognition around this, or are we broadly speaking about the forebrain, for instance, the hypothalamus being in the forebrain?
So yeah, it's interesting. So the thirst circuit, for whatever reason, is mostly in the forebrain. So the neurons so we talked about the NTS and the area postrema being important for hunger and signals from the gut. The area postrema is a circumventricular organ, meaning it's outside the blood-brain barrier. There's only a couple of these in the brain. The neurons that control thirst are located in the two circumventricular organs in the forebrain. One is called the subfornical organ. The other one is called the OVLT, but they're just acronyms. But so why it evolved to have the thirst neurons more in the forebrain and the neurons that sense nutrients more in the hindbrain is a little bit unclear. And so there is definitely an element of learning. But a lot of this is those neurons are also just directly sensing the blood and sensing changes in both the concentration of salt in the blood and then also hormones like angiotensin that drive thirst.
I was going to ask you this earlier, but it seems appropriate to ask now. A colleague of mine at Stanford in the psychology department, Dr. Ali Crum, who studies mindsets, has done some interesting experiments where people are told that a given milkshake is calorically dense. Other people are told that a milkshake is calorically sparse. Both groups independently consume the milkshake, and then they measure things like hormone responses in the bloodstream that are associated with satiety. And what she finds is that even hormone responses to the same shake, meaning the same amount of calories, fat, sugar, etc., can be significantly modulated based on what we're told. Mm-hmm. And it extends into some other, perhaps even more interesting areas, in my opinion, whereby if people are told that, let's say, a given meal that has a small piece of fish, a serving of vegetables, and a carbohydrate is, yes, perhaps a little bit calorically sparse compared to what one would normally eat at a given meal, but they're told this is a highly nutritious meal. This is good for you.
Yes.
Then just that mere knowledge can drive more satiety, better feelings about the meal, even, I believe, I have to double check on this, but as I recall, a heightened sense of it tasting really good. So humans are very susceptible to in this case, the either inaccurate, in the case of the milkshake experiment, or accurate descriptions of food, meaning they shape our perception of whether or not something is good for us, tastes good or not, and whether or not it leads to more or less satiety. Mm-hmm. And I think this is important given the obesity crisis, to say nothing of these drugs that are coming out, whereby people often associate dieting with deprivation and pain. But if they understand that certain foods are nutritious, that can at least partially offset some of the pain of caloric restriction. What are your thoughts on that?
Yeah. Well, one thing I've been talking about is how a lot of these circuits are anticipatory. They're making predictions. They're trying to estimate what's happening in the future. And I talked about how these AGRP hunger neurons, how they can sort of see the food or get input about the sight, smell of food, and in that way predict how many calories the mouse is going to eat. But, this is a mouse, right? This is all based on a mouse. And a mouse has, 1,000 times fewer neurons than you do as a person, right? So the computational capacity that the human brain has to make these predictions is just vast compared to these mice. And these mice are already doing amazing things, right? So when you think about then, what is the human brain able to do in terms of anticipating changes in nutritional state and how information that you're given can change the expected physiologic outcomes? I mean, you're right. I mean, there's just this whole other element that is very hard to study because it's happening in the brains of humans and we can't do these kinds of experiments. But I'm sure that's very important. I mean, so I talked a little bit about these flavor nutrient conditioning experiments. These are the experiments where essentially an animal learns to consume a certain flavor because it learns it's going to be associated with nutrients later. Sort of the paradigm for how you learn to consume bitter vegetables because they're good for you and you get nutrients. So people have also done those experiments in humans, and that does work. But what they've discovered is it's very sensitive to what you tell the humans about the thing that they're going to consume. So if you put nutritional labels where you show the different numbers of calories, then basically they sort of adjust their expectations and nothing happens. So it really has to be that sort of, it's very sensitive to what information you give them before the experiment happens. So I think that's an example of that kind of thing.
Without any pressure for it to be prescriptive, how do you approach eating, given the knowledge that you have about food? I like to assume that you can sit down to a meal and not think about your AGRP neurons too much or any of that. But given that you have deep knowledge in this, has it shaped kind of how you think about food cravings, your own? You don't have to reveal what those are, even if they exist. How you observe the eating behavior of others, and yeah, how has knowledge shaped your feeding behavior?
Well, I try not to think too much about my AGRP neurons when I'm eating. Because
I would hope.
I think it gets. I think the circuitry is so complex, and we're just beginning to see what's happening. So I wouldn't use that kind of information at this stage, and we're just beginning too prescriptively. But I think there is a set of basic recommendations from physiology and neuroscience, very simple things you've probably talked about with people on your podcast before, for sort of shaping your diet to be healthier, to limit food intake. So one we've already talked about is limiting consumption of ultra-processed food, eating more whole foods for lots of different reasons, because they're more satiating, because they don't have this sort of engineered palatability that causes you to overeat. Another big one, which I'm sure you've talked about with some of your guests, is protein consumption, making sure you get adequate protein consumption, both because there's this concept of protein leveraging. So if you don't eat a minimum amount of protein, that's going to cause you to eat more calories just to try to achieve that minimum amount of protein. Also just because protein's more satiating, and also because there's this idea of thermic effect of food. And so you basically burn more calories metabolizing protein than sugar or fat.
How about consumption of fluids during meals? I've heard it said before that we're not supposed to consume too many fluids because it's going to dilute the enzymes that allow us to digest our food. I've heard other people say that's complete.
I think that's
a myth.
That's a myth. I think drinking water. So humans don't have a perfect capacity to determine whether they're hungry or thirsty, and so drinking water will ensure you're not eating because you're because you're thirsty. And there's no idea of diluting it. I don't think that. And distension itself, even though water provides a very limited distension signal, the expansion of your stomach and intestines is one important way that you terminate feeding. And so there is some component of that where you can get distension just from drinking water.
I blurted out. Interesting, because I didn't realize that fluid consumption only provides a limited signal for distension.
Well, it's not fluids, it's water. And so the idea is that you can fill your stomach up with fluids, but the rate at which fluids empty out of your stomach depends on their calorie content. So basically, if you drink water, it empties very rapidly into your intestine and then goes through your intestine and is gradually absorbed. If you drink something like a glass of orange juice, it will empty much more slowly. And if you drink something that's really high in fat, really high in calories, it will empty extremely slowly over hours. And that's because there's a negative feedback loop from the intestine that controls gastric emptying. So as those first nutrients leave your stomach and enter your intestine, that produces hormones that go back and then slow down the rate of gastric emptying. And the purpose for this is that you don't want nutrients entering the intestine too fast. That's really unsafe. It feels very unpleasant. And it's just your intestine can only metabolize nutrients so fast. And so if there's calories, then it slows down gastric emptying a lot, but water just kind of goes through.
What a beautiful system. Like there's regulation at every point. Right. Hypothalamus, brainstem, gut, rate of emptying based on the difference between water and orange juice. It's just awesome.
Yeah, and that's part of the reason I think it's so hard to outsmart the system, right? Because, these neurons are making predictions based on the sight and smell of food, but then the gut is doing its own thing. It's calculating it separately and relaying that information. So at every step, there are these checks. Basically, they're just confirming that what you thought happened the first time is actually what's really going on. And so, which just makes sense because it's so important for survival. These homeostatic systems are the product of so much natural selection.
Which I think at least partially explains why 1,000-fold increases in peptide hormones like GLP-1 are required to see significant long-lasting. changes in weight
Exactly.
because the system is so strongly regulated.
Exactly.
It's hard to beat homeostasis and hard to beat it safely. But it sounds like you're more or less optimistic about where that whole field of let's call it anti-obesity drugs is headed.
I'm very optimistic. I think that you couldn't have asked for more so far at this stage with these GLP-1 drugs. Incredible weight loss, unexpected health benefits, really safe as far as we can tell. It's always possible that some new side effect will emerge, but these drugs are in millions of people, and they've been in a lot of people for a long time now, and nothing seems to have shown up. So I'm very optimistic. And I think even beyond that, just now that the pharmaceutical industry is reinvigorated to investigate this question, there's so many different, in 5 years, people have so many different options. It won't just be Ozempic or Mounjaro. There will be 10 different drugs that they can choose from that have slightly different side effect profiles, slightly different efficacy, perhaps used for people with slightly different metabolic conditions. And so it'll really be a whole palette of medicines you can take that will adjust your physiology and hunger.
And it's amazing how well it squares with the understanding of the basic biology, and that's a perfect opportunity for me to really just say what is in my mind and clearly in the minds of everyone listening and watching, which is thank you so much for this absolutely encyclopedic and exceptionally clear explanation of feeding and thirst and salt regulation and these new drugs that are, in everyone's minds and everyone's hearing about. I've learned so much today. I know everyone else has. You run a incredible laboratory. I've tracked your career for a very long time. Every paper is spectacular, and you're in a very competitive field, and you've contributed in enormous ways to our understanding of these important processes. And I don't just say that as a formality. I know that to be true, given that we, are from the same field and have known each other for a long time, and I'm familiar with your work at a deep level. Today has just been an absolute privilege and a gift to learn from you, and I know everyone feels the same way. So thank you for taking time out of your busy research schedule and the other important areas of your life to come here and educate us all. I learned so much basic and practical knowledge, and I know everyone else did as well. Thank you so much.
Thank you. This has been really fun. I'm really glad we had a chance to do this. We talked about some of my favorite topics, so it's always a pleasure. And to talk with another neuroscientist about these things is fantastic. So,
all right, well, please come back again. Meanwhile, thanks for everything you do.
All right, thanks.
Thank you for joining me for today's discussion with Dr. Zachary Knight. To learn more about his research or to support his laboratory's work, please see the links in the show note captions. If you're learning from and/or enjoying this podcast, please subscribe to our YouTube channel. Please also subscribe to the podcast on both Spotify and Apple by clicking the follow button. That's a terrific zero-cost way to support us. Also on both Spotify and Apple, you can leave us up to a 5-star review. Please also check out the sponsors mentioned at the beginning and throughout today's episode. That's the best way to support this podcast. If you have questions for me or comments about the podcast or topics or guests you'd like me to consider for the Huberman Lab podcast, please put those in the comment section on YouTube. I do read all the comments. If you're not already following me on social media, I am Huberman Lab on all social media platforms. So that's Instagram, X, formerly known as Twitter, Threads, Facebook, and LinkedIn. And on all those channels, I cover science and science-related tools, some of which overlaps with the contents of the Huberman Lab podcast, but much of which is distinct from the contents on the Huberman Lab podcast. So again, that's Huberman Lab on all social media platforms. And if you haven't already subscribed to our Neural Network newsletter, our Neural Network newsletter is a zero-cost monthly newsletter that includes podcast summaries, as well as brief 1 to 3 page PDFs of what we call protocols. Those protocols cover everything from learning and neuroplasticity to dopamine regulation to improving your sleep. We also have a foundational fitness protocol that details cardiovascular training and resistance training sessions, right down to the details. Again, all of that is available completely zero cost. You simply go to hubermanlab.com, go to the menu tab, scroll down to newsletter, and enter your email. And I should point out that we do not share your email with anybody. Thank you once again for joining me for today's discussion with Dr. Zachary Knight. And last but certainly not least, thank you for your interest in science.